Hi everyone, this is Katyani and welcome to our channel. Me and Tina will be guiding you through IB biology, and the next four videos you will see are based on chapter 3, which is genetics. So to start off with the first section of genetics, which is genes, we have a bunch of learning outcomes that we have to go through. So this might take a while. So the first thing we have to start with is, what is DNA? DNA is essentially your blueprint, which codes for and determines your characteristics as well as the characteristics of every other organism. It is packaged and organized into structures called chromosomes. And after that, after chromosomes is something called a gene, which is a sequence of DNA that encodes for a specific trait. And the position of a gene on a particular chromosome is called a locus, and the plural of that is loci. So these are some definitions that you need to know in case they come up. So this is a visual image of that. So you can see that the gene is on the chromosome, and that is known as a loci, and that codes for specific traits such as blue eyes or brown hair or something like that. Now, alleles are alternative forms of a gene that code for the different variations of a specific trait. So since alleles are alternative forms of one gene, they possess very similar gene sequences. So as you can see in this picture, one person has different alleles or different genes that they inherit from their father and mother. And this is known as a Punnett square, which determines what the child is going to have in terms of their genes. So a gene mutation is a change in the nucleotide sequence of a section of DNA coding for a specific trait. So here you can see that if a DNA has no mutation, it's TTC. If it has a silent, then it's TTT. If it's missense, it's TCC, and if it's nonsense, it's ATC. So this part you don't really need to know for your exam, but just know that a mutation is a change in the nucleotide sequence of a section of DNA. Okay, so the disease that mostly always comes up in the exam is sickle cell anemia, and you need to know that it is a disorder that is caused by a gene mutation, and this happens where a single base is changed in the sequence. So what is the cause of anemia? It's basically, it results from a change to the 6th codon in the chain of hemoglobin. So the different causes can be DNA, which changes from GAG to GTG on the non-coding strand, and CTC to CAC on the coding strand. Another cause can be mRNA, and this is when the sequence changes from GAG to GUG on the 6th codon position. So the difference between this is that in DNA it's GTG and in mRNA it's GUG. Make sure you know that. And the next cause is polypeptide, where the 6th amino acid for the beta chain of hemoglobin is changed from glutamic acid to valine. So these are very important. They will get you the 3 marks. So this is just a visual aid for what I just said. So CTC to CAC, GAG to GUG, and glutamine to valine. What is the consequences of sickle cell anemia? Basically, what happens is that it alters the structure of hemoglobin, which causes insoluble fibrous strands. Therefore, your hemoglobin will not be able to carry oxygen as effectively, which causes an individual to feel tired all the time. Also, the fibrous hemoglobin strands changes the shape of the red blood cell to a sickle shape, as you can see in the diagram, in the picture. And the sickle cells may form blood clots within the capillaries, blocking the blood supply to vital organs such as the lungs, liver, kidneys, and this causes health issues. The sickle cells are also destroyed more rapidly than normal cells, leading to a low blood cell count, which again makes you feel tired. Now, what is a genome? A genome is the totality of genetic information of a cell, organism, or organelle. This includes all genes as well as non-coding DNA sequences such as introns, promoters, short tandem repeats, etc. And you don't need to know this, but just for general knowledge, the Human Genome Project was an international venture that was just established to sequence the genome. And finally, you need to know that these are the gene comparisons between different species. You do need to remember these numerical values, so I would recommend just noting them down and saying them every day so that when the exam comes, you don't blank out. And that is all for section 1, so I hope it helped.